Our next presentation is also being given by Soraya Bertioli, entitled Mobilizing the Multiple Resistance of Arachis neopolyploids for Peanut Improvement. Soraya, take it away.
Great, thank you. So we were talking about Arachis neopolyploids earlier, and I'm pleased to present our star of the day. Arachis neopolyploids has been very important in finding resistances. So there is a work that was done by Michelotto, is a group in Brazil, in São Paulo. They made lots of bioassays, a lot of evaluations with a whole group of wild species. And they found that the different accessions of Arachis neopolyploids, even from different regions of the country, even adapted to very, very different environments, had resistance to these four diseases, fungal diseases here: late and early leaf spot, scab, and rust. In the work we've done, we've paid a lot of attention to Arachis V10309, and we are starting to work with accessions from the coast as well. So I'm going to share a little bit about the work we've done with these, especially 10309. So this accession was found in a place similar to what is shown here. Is in the west of Brazil. It's called Rondonópolis. Is a region that in the 70s and 80s, there was a big migration there because people from the south were given or sold land, very subsidized by the government, to plant soybean. So that's big soybean country. So obviously it's great for the economy, etc., for food, but of course the wildlife, the plants, we suffered a lot with biodiversity. We lost a lot of it. So Arachis neopolyploids was collected in an area similar to this, which was just on the outskirts of this city called Rondonópolis. This city exploded. It really exploded with migrants from the south. And by 2004, it looked like this. And if you see here, there are some few remains of the accession that had been collected almost 20 years before. And this is what it looked like 2 years ago. So no Arachis neopolyploids anymore. Well, not that accession. So luckily, this was collected. It was saved in different gene banks, and it is available at the USDA. So we're very lucky to have the USDA collection curated, preserved here in Griffin. So the Arachis neopolyploids, we've shown this slide before, and this was also the theme of yesterday's talk from Kate Costello, our student. Arachis neopolyploids has strong resistance, and we found at microscopic level, for late leaf spot. You see here germinated spores and germ tubes in Arachis hypogaea, and you see a lot of proliferation and presence of a dense net of germ tubes, whereas in Stenosperma there is hardly any penetration. And the same happens with rust. It's also very resistant to nematodes. We did this work in Brazil quite a few years back. We tested for all these 5 different species of nematode, including Ditilencas africanus, which has a completely different infection mode. It's a stem nematode. And Arachis neopolyploids was resistant to all of them, a little less to Hapla, but much more than Arachis hypogaea. We concentrated our efforts in Arachis RNA race 1, which is the one that is the most important in the United States. And again, we find that the resistant is completely different from Arachis hypogaea, that you have penetration here, and in 32 days you have lots of feeding sites. And in Stenosperma, we hardly have any penetration. And when we do, there's necrosis, we have hypersensitive response around the pairs of penetration. So what we did with this, we made tetraploids, we made it available by crossing it with a B diploid, actually crossed initially with a K, which is bachysocoi. Made sterile hybrids, treated with colchicine, let it grow, repeated, let it grow, repeated. And eventually we got successful, which we call BAT10. So just to give you an idea, from these V10309, we have 5 allotetraploids, 3 of which were released last year between our groups and Pega's group. And there are 3 from the coastal region, and 1 was released last year. It's just to show you what they look like. They have very wild traits. They have very small seed. They have very wild looking like seed. But very quickly, from the BCA1 onwards, you can recover the seed size and also yield. So this is going to be shown to you by Carolina Balen in the next talk. I just want to give you an example. One of them is called Valstan. This was produced by these particular ones were produced by Dongjin Gao, who was a postdoc in the lab for a while too, and was released this year by Juliette Chu, some that they produced over there. So this is a very interesting case, very prolific. It was very easy to duplicate. It was very easy to cross, and we had many, many events of tetraploidy. And these events, there was a lot of diversity. You have different flowers, completely different looking flowers from the different events, different looking leaves. So the diversity was enormous. But one thing that was the same was resistance. So all these events that we tested were resistant to early native spot. And again, why do we want diversity? Again, from the wise words of Tim, we need to be proactive rather than reactive. And from what Albert said earlier, the coyote is always waiting. The coyote is always hungry. So we want to be on the lookout. We want to be sure we have enough bullets for different races of fungi, for different species of fungi or other pests, for different constraints, for different environmental changes. And we want to have enough allelic diversity to be prepared. So this is probably going to show a lot of this slide. This is the wild peanut lab pipeline, which starts with the wild species collection, seed exchange, seed bank curation that is done, this is done by people who preceded us and we're very grateful for. And we start pretty much here in the characterization of the resistances. We make the primary hybrids, tetraploidize. So we have a mag stand that we have recently tetraploidized. We cross with peanut. So these are the stages. These are the stages where each of these allotetraploids is. And this is the most advanced one that we have. These being planted in different regions. This combines root knot nematode, late leaf spot, and possibly TSWV resistance. And we hope to very soon release some improved varieties with segments of Arachis stenosperma. With that, I'd like to thank the whole Wild Peanut Lab and our collaborators, obviously, and to remind you that we are hiring postdocs and technicians. So thank you very much.
So we have quite a bit of time here to ask some questions. So please post your questions to the Q&A, and then I'll speak to the group. Any questions for Soraya? So we do have one question that just popped up from Jiangping Wang. How stable is the genome of the synthetic tetraploid derived from A. stenosperma? Any genome losses? any recombination between A and B subgenomes?
Right. Well, you know you're talking about, that's a good question. Yes, there is recombination. There is quite a bit of recombination. We found that, for instance, there in the ipaënsis duranensis, which is from the progenitors of peanut, we find so much recombination that when we genotype one of the lines, we don't see the one of the genomes gets shut down. So we only see markers for one of the genomes. So there is mostly tetraploid, tetrasomy recombination at the ends of the chromosomes. So we see that quite a lot. And some of the progeny may not be so stable. So these are things that we are learning as we produce these materials. And we used to treat all the events, all the tetraploids from one cross as one. And now we see that we have to treat each event and also each generation, label it well so that we know what we're talking about, because there is difference and there is instability, which is also fun because also enhances the possibility of bringing different allelic combinations.
I guess just a quick follow-up on that. When you're looking at the amphidiploid route versus say the hexaploid route, do you tend to see more of those recombination events between subgenomes in the hexaploid? You know, if we're considering the Carnaceae introgressions versus, you know, maybe some of these from Stenosperma and the backcross? We
don't know. That's the true answer. We don't know, because we don't have a very good control over the hexaploid route. That was a question that Tom asked me some time ago.
Mm-hmm.
If we had examples of each of the generations of the hexaploid that we could genotype nowadays, we'll be able to compare. But we don't, so we don't know. I think the hexaploid is less stable. I mean, the nature of it is instability. We want it to be unstable, so it's so unstable that the chromosomes get kicked out. So even looking at the big picture, the hexaploid seems to be less stable to me, but that's just a wild guess.
Yeah. Our next question comes from Steve Brown. Having the source of resistance, identifying the QTLs and markers that help us pinpoint the resistance for breeding is very important. How important is the next step of identifying specific genes and mechanisms of resistance?
Well, it's always important to know what we are looking at. I think knowledge is always excellent, and it's always good to know how some things work, so that when it doesn't work, we know what went wrong. Okay. We are more focused on producing the materials, understanding how they work at the moment, understanding the stability or the instability of it. But yes, learning how they work, knowing about the genes, is the next step, definitely.
And then I guess the final question here we have is from Josh Clevenger. Thank you to the Wild Peanut Lab for providing the genetic tools for the next generation. Within the pipeline, I wonder, do you see certain introgressions always coming through the same physical size? Within a population, is it possible to select the smaller introgressions? I wonder because sometimes segments are very large, and sometimes with like the Carnaceae segments, they can be much smaller.
Oh, absolutely, absolutely. That's a very good observation, Josh. We're actually doing that with the last one. Let's see, yeah. So with this population here, this BC4 population, we actually now, it's the work of a new PhD student, Emil. So she's, breaking down some of these segments, and we want to see what's the smallest possible size that we can still confer resistance to root knot nematode, and still have confer. So yes, I think it's important to have the smallest segment possible, because obviously we can have linkage drag with the wild. And thanks for the comment, Josh. You were certainly part of these of bringing the tools to the community, and you still are. So thank you.